Welcome to the lectures on multivariate twin modeling in the International Statistical Genetics Workshop. These 5 lectures are given by myself, Connor Dolan, and my colleague Dirk Pelt. We're going to present 5 PowerPoints, and the first one is about the step from the univariate to the bivariate model, twin model. The second PowerPoint lecture is about the step from bivariate to multivariate. Lectures 3 and 4 are about the independent pathway model, and lecture number 5 is about the common pathway model. So before we start with the step from univariate to bivariate, I have 2 preliminary slides. This is just an overview of the notation that we use in these 5 lectures. So you could stop this show now and take this in. And the second preliminary slide concerns 2 ways of representing a covariance. I'll be using both, so I thought I'd devote a little bit of time to this just to make sure it's clear. So you see 5 covariance matrices in this slide. The top left one is the 2x2 covariance matrix of phenotype 1, phenotype 2. The diagonal elements are the variances, of course. The off diagonal is the covariance. So sigma pH1, pH2, that is one way to represent the covariance. Now in the first row, the middle matrix in the first row, that shows you exactly the same covariance matrix. The only difference is that we're representing the covariance between the first and the second phenotype differently. Yeah, so it's the standard deviation of the first phenotype times the correlation between phenotype 1 and phenotype 2 times the standard deviation of the second phenotype. So that's the second way to represent a covariance. Now the matrix top right, that's just a numerical example, and then under that, the second row, you can see the correlation matrix. So all the way to the right, you can see that the correlation is 0.5. And how do we calculate that correlation? Well, you can see that if you look at the second row matrix on the left. Yeah, that's just an aside. The important result is shown at the bottom. The covariance is either represented as sigma pH1, pH2, or standard deviation of phenotype 1 times the correlation times the standard deviation of phenotype 2. Okay, so what we're going to start with briefly is the univariate twin model. I just want to remind you of this because we're going to use that to step up to the bivariate twin model. So this is an example of a univariate ACE model. The path diagram shown on the left depicts the model in the variance components parameterization. So the parameters of this model is var A, the variance of A, var C, and var E. And on the right, you can see the expected covariance matrix in the MZ twins, the monozygotic twins, and at the bottom, the dizygotic twins. Under those matrices, you can see that I've expressed the standardized additive genetic variance, the narrow sense heritability, which is the additive genetic variance divided by the total variance. And you can express that with 2 different notations, depending on how you want to express the variance of A, yeah, var A or sigma A squared. This is again the univariate ACE model, but now the path coefficient representation, parameterization, I should say. So that is, this model has 3 parameters, lowercase a, lowercase c, lowercase e, and those 3 parameters make up the covariance matrix in the MZs and the DZs. And then at the bottom of those 2 matrices, you can again see the narrow sense heritability, standardized additive genetic variance in the 2 different notations. Okay, so that was just to remind you of the univariate twin model. What we're going to do now is we're going to move on to the bivariate twin model. We're not going to consider twin data directly. First, we're just going to focus on 2 phenotypes. The run example in actually the first 4 lectures are the skinfold data. So these are measured skin folds at given locations on the body, and this data set, the skinfold data, that is available in the R library called UMX. And at the top you can see Moscovich, that is the source of these data. Okay, so we're going to consider skinfold data, and we're going to start with biceps and triceps. So the measures are measures of skin fold taken at the biceps and the triceps. Those are the places, the locations shown in purple. So here are the results shown in the part diagrams of the univariate analysis of the biceps skinfold data and the triceps skinfold data. So what we're doing, what I'm presenting to you, is the decomposition of variance. The variance of biceps is 13.38, shown in red, in the covariance matrix top right. The variance of triceps is 11.53, again shown in red. And these results show you the decomposition based on the univariate twin model of these variances. In the part diagrams, you can see I've included the raw variance estimates and also the standardized variance estimates. So, for instance, the contribution of additive genetic effects to biceps is 37.6% of the phenotypic variance. Okay, so this gives you a decomposition of the variance, but we know that biceps and triceps skinfold data, that they co-vary, because that's shown in the covariance matrix. We can see that the covariance is equal to 11.16, and that the correlation is 0.899, shown in parentheses. Okay, so based on the univariate analysis, we know a thing or two about the composition of the phenotypic variances, but we don't know anything about the contributions of E, D, and A to the covariance. So what we're going to do is we're going to go from the univariate model to the bivariate model to address that question. What are the contributions of E, D, and A to the phenotypic covariance? Okay, so what you see here is in the part diagram the model that we actually want to fit using variance components. So biceps and triceps co-vary, and we assume that they co-vary because the E effects on biceps are correlated with the E effects on triceps. So the E effects for biceps, triceps, they co-vary, and the parameter, the covariance between those effects, is called cov EB/ET, shown in the part diagram, and the same applies to the D and the A. Additive genetic effects on biceps co-vary with additive genetic effects on triceps. So we're explaining the phenotypic covariance between biceps and triceps by estimating the covariance between the E, D, and A effects. Okay, so on the right hand side of the slide you can see the covariance matrix in symbols and numerically, and in blue again the covariance, and under that you can see how we are representing the phenotypic covariance matrix. So we're going to decompose the variances into 3 parts, and we're going to decompose the covariance into 3 parts. And this, so what are the parts? Well, it's the covariance between the A effects, the D effects, and the E effects. Sigma AB/AT, sigma DB/DT, and sigma EB/ET. Okay, so we're going to go from a decomposition of 1 variance to the decomposition of 2 variants and a covariance all at the same time. Okay, so the covariance matrix shown here top left, that's going to be decomposed into, so sigma PH decomposed into sigma A, uppercase sigma A, uppercase sigma D, uppercase sigma E. And that's what this slide shows you, the decomposition of the phenotypic 2x2 covariance matrix into its 3 parts. And then at the bottom is the result of the sum. When you apply element by element matrix addition, you get the model for the phenotypic covariance matrix. Okay, so far I've discussed the covariance matrix for triceps and biceps, but I've not referred to twins. What we're going to do is we're going to use the classical twin design to obtain estimates of sigma A, sigma D, and sigma E. So that requires us to expand the model from a 2x2 matrix to a 4x4 matrix, two phenotypes measured into twins. So that's what you see here. The phenotypic covariance matrix for the MZs, that's top left, and for the DZs, bottom left. Okay. What I'd like to do before we actually fit the model is represent the model, the bivariate twin model, as a path diagram, using variance components parameterization. That's what you see in this slide. And you can see that this is a pretty complicated slide. If you remember, the univariate twin model is easy to take in, but this includes many double-headed arrows. This is the path diagram for the MZs. This is the path diagram for the DZs. And you'll note, what are the differences between the two? Well, the coefficients associated with the A and the D. We went from 1 and 1 in the MZs to, as you can see by inspecting the coefficients in this slide, 1/2 and 1/4. Anyway, again, this is pretty complicated. So what we're going to do is we're going to break it down into its parts. So what we're going to start with is just the univariate twin model for biceps. And that's the path diagram shown here in the variance components parameterization. So this is the model for biceps univariate. And under that, you can see the DZ covariance matrix, phenotypic covariance matrix, that's 4x4. And they're showing you what this part of the model contributes to that 4x4 covariance matrix. So what is it contributing? Well, the variance of biceps in twin 1 and twin 2, diagonal elements, and then the covariance between biceps in twin 1 and twin 2, the off diagonals. This is the first univariate. This is the second univariate model, because now we've gone from biceps to triceps, but exactly the same narrative. What does this contribute to the 4x4 matrix? Well, the variance of triceps in twin 1, in twin 2, and the covariance as shown at the bottom of the slide in the matrix. And the double-headed arrows shown in orange have highlighted their role in the matrix in the bottom. They're contributing to the covariance. Okay, so that was univariate model 1 and 2. What we're now going to consider is the covariance between biceps and triceps within twin members. So this is not twin 1, twin 2, or this is twin 1 and twin 2, but we're only considering the covariance and the variances within twin members. So what this shows you is, what is this contributing to the matrix? Well, it's contributing the variance of biceps and the variance of triceps, the diagonal elements, and it's contributing the covariance between biceps and triceps in the twin 1 members and in the twin 2 members. So you can see that at the bottom. Okay, the next step is we take the model in the previous slide, and now we're going to start adding the double-headed arrows. Step number 1 is to connect the AD of biceps in twin 1 to the AD in biceps in twin 2. So what we're doing is, by adding these orange double-headed arrows, we're adding off-diagonal elements in the matrix shown at the bottom. So that is the covariance between biceps twin 1, twin 2, and the covariance between triceps in twin 1 and twin 2 in this slide. So at this point, we've added the expected AD covariances between twin 1 and twin 2. I should expect them based on the univariate models. The final thing to do is to connect the A of biceps in twin 1 to the A of triceps in twin 2. That applies to the A and the D influences. So these are the cross-twin, cross-phenotype covariances, and they contribute the elements to the 4x4 matrix shown at the bottom in green. So that is, for instance, you can answer this question by reading off the correct cell in the matrix at the bottom. What, according to this model, is the covariance between triceps in twin 2 and biceps in twin 1. Yeah, that's element. The fourth row, the first column, answers that question. Let's return briefly to the part of the model we're actually interested in, that is the decomposition of the covariance matrix of biceps and triceps, represented in a path diagram in two parameterizations. On the left, variance components; on the right, path coefficients. Generally, we use variance components to fit the model, especially if we want to test variance components. But we can represent the results once we have them using path coefficients if you like. And I do like. I think it's a nice way to represent the results. At the bottom, you can see two matrices. This is sigma A, so this is the additive genetic covariance matrix in this model, associated with each parameterization. On the left, you can see that the covariance between biceps and triceps, the additive genetic covariance is sigma AB AT. Yeah, that is the representation in the variance components. On the right, that covariance is modeled as AB times the correlation rho AB AT times AT. Yeah, so that's the two different ways of representing a covariance. Okay, now we have this model in place. What I'd like to do is I'd like to fit it to the skinfold data. So let's take a look at a numerical example. Okay, so this is a busy slide because it contains the summary statistics in the MZ and the DZ samples. So we have a sample of 214 monozygotic twins and a sample of 181 dizygotic twins. The first row at the top are the covariance matrices. Shown in blue are the covariance matrices within twin members, so twin 1 and twin 2. And shown in red are the covariances between twin 1 and twin 2. Under the first row, the second row shows you the correlation matrices based on the same data. Okay, so we can visually inspect the correlations to get an idea of what model might be suitable for these data. And one thing we see immediately, as shown in the green boxes, you can see that the MZ correlation is about 0.78 and 0.80. So 78 and 0.80, and the DZ correlations are about 0.32 and 0.27. So 2 times the DZ correlation is smaller than the MZ correlation, and that suggests that an ADE model might be suitable. So what we're going to do is we're going to fit a bivariate ADE model. What are the expected covariance matrices? They're shown at the bottom. Yeah, and I've adopted the color coding in these two matrices. Yeah, so blue is the covariance matrix 2 by 2 within twin members, and red is the covariance between twin members. Okay, so I fitted this model, and what you see here are the results. And I'd like to go over these carefully and discuss specifically the covariance between biceps and triceps. Yeah, so the decomposition of the variances, that is relatively simple, and we've already on my earlier slide I showed you that decomposition in the path diagrams for biceps and triceps separately. So what we're going to do is we're going to look at the decomposition of the covariance. So what is the covariance? It's 10.52. Yeah, shown in red. That is the expected covariance based on fitting the bivariate ADE model. Now that 10.52 is decomposed into an A covariance, a D covariance, and an E covariance. And those are shown in green. So 2.78, 5.91, and 1.87. Okay. Now, what I can do is I can represent the covariance, the antigenetic covariance, as 2.78, yeah, shown in green, or I can represent that alternatively as the additive genetic standard deviation times the additive genetic correlation times the additive genetic standard deviation. Standard deviation, additive genetic standard deviation of biceps, additive genetic correlation times the additive genetic standard deviation of triceps. So that's what you can see on the right in this slide. Yeah, so representing the covariances, the 2.78, 5.91 and the 1.87 in two different ways, just as a covariance and also involving the standard deviations and the correlations. Okay, so that is two different ways to represent the covariance, which I discussed earlier. Now, a question we can ask is, so if the phenotypic correlation, as the phenotypic covariance is 10.57, how much of that is attributable to additive genetic effects? And to answer that question, one way to answer this question is to express the contribution of A, the 2.78, as a proportion. So if I divide the 2.78 by the total phenotypic covariance, that gives me 0.26. So 26.3% of the phenotypic variance between biceps and triceps is explained by additive genetic effects. So this is proportions on the one hand, and on the other hand, two ways of representing the covariance. Okay, so the previous slide showed you the results numerically, just in tables, sigma A, sigma D, sigma E. What we see here are the results represented in the path diagram. On the left, the variance components, and on the right, the path coefficient representation. These are equivalent representations. What we can also do is we can take the path coefficient representation and standardize the variance components, and that gives you a representation which is even easier to take in. Let's see that in the next slide. In this slide, on the left, you can see the path coefficient representation of the results that we saw in the previous slide. But I've added to that the path coefficient representation of the results where the variances of the phenotypes are standardized. Yeah, so the path model on the right is easier to interpret because I can see directly what the contribution is of additive genetic effects to biceps. It's 37.4%. Yeah, that's the narrow sense heritability associated with biceps. So the difference between these two is the following. In the figure on the left, the A, D, and E latent variables are standardized, but biceps and triceps are not standardized. Yeah, so the 4.729, yeah, that's the additive genetic variance associated with bicep. To evaluate whether that's large or small, you'd have to divide that by the total variance of biceps. Yeah, and once you do that, you get the narrow sense heritability, which is readily interpretable. So what I've done is on the figure on the to the right is I've calculated the standardized variance components and I've inserted those as the path coefficients. They're easier to interpret. So what is the difference? Biceps and triceps are not standardized on the left. ADE on the left are standardized. In the figure on the right, ADE are standardized and the phenotypes, biceps and triceps, are also standardized. That makes it easier to interpret. The standardization pertaining to biceps is shown at the bottom of the slide. Yeah, so what am I doing? Dividing the variance components for A, D, and E by the total phenotypic variance, and that gives me the standardized variance component. Okay, this is the last slide, and this slide has two questions for you to think about. And question one is this. Take a look at the path diagram top left. Okay, the contribution of A, AB, AT to the phenotypic covariance between biceps and triceps depends on three parameters: AB, rho, AB, AT, and AT. Yeah, so that's the alternative representation of the covariance between the additive genetic covariance between biceps and triceps. And the question is, suppose I told you, I show you this model, top left, and I say, well, the correlation between AB and AT, shown as rho AB, AT, that correlation is very high, it's 0.9. Does that mean that additive genetic effects are making a large contribution to the phenotypic covariance? Yeah. So if the correlation between the A's is very, very large, does that mean that the A's are making a large contribution to the phenotypic covariance? That's question 1, and question 2 is more complicated and more subtle. So this is certainly something to think about. And the question is this: why can we not estimate rho ABAT? That's a correlation between the A's. Why can we not estimate that parameter accurately if A, B, and or AT are relatively small? Yeah, so the A, B, and AT are the part coefficients. If they are relatively small, one or the other or both, why can we not estimate the correlation between the A's accurately? Okay, so for instance, suppose that AB accounted for 3% of the variance in biceps and AT accounted for 40%, that's 3% versus 40%. In that situation, it could be difficult to estimate rho ABAT accurately. Okay, and why is that? This is a question that we're going to return to in the practical. This concludes the first PowerPoint presentation.